Today, we are diving into a really high-stakes medical mystery. We're talking about cracking the code to protect the brains of some of the most vulnerable people on the planet, newborns.
So check this out. There's a treatment out there that can literally save a newborn's brain from permanent damage. I mean, that's incredible, right? But here's the catch. It only works about half the time. It's basically a coin flip. And you can imagine, for doctors, for parents, that 50-50 chance is just agonizing. So the big question researchers had to answer was, why? Why does it save some babies, but not others?
Okay, so the condition we're actually talking about here is called neonatal hypoxic-ischemic brain injury, or HI brain injury for short. It's a really devastating type of brain damage that can happen when a baby's brain doesn't get enough oxygen and blood right around the time they're born.
The standard treatment, the one doctors turn to, is called therapeutic hypothermia. And it's pretty much what it sounds like. They carefully cool the baby's body down. The idea is to protect the brain, to stop permanent damage from setting in. But, and this is the huge but we started with, it only works for about half the babies. The other half, they just don't get that protection. And for the longest time, nobody could figure out why.
So to solve this incredibly tough puzzle, researchers realized they needed a new kind of detective. So they brought in the big guns, a digital detective, an AI they named Cosmos. Its job? To go on a hunt for clues buried deep inside the body's own biological data.
Cosmos's first assignment was to dig into two massive piles of evidence. This data came from a mouse model that mimics this exact type of brain injury. I mean, we're talking about sifting through thousands and thousands of data points, all to find that one hidden pattern, the thing that separates a brain that's protected from one that isn't.
Now, the AI wasn't just looking at one type of clue. It had two very different kinds of evidence to work with. Let's break it down. The first set is called metabolomics. Think of this as the "what's happening right now" report. It's a real-time snapshot of all the chemical activity inside the cells. Then you've got the second set, transcriptomics. This is more like the "what's the plan" report. It's all the genetic instructions the body is sending out to respond to the crisis. So you've got two different languages, both trying to tell the same story.
And right away, Cosmos found a huge lead. What you're looking at here is a comparison of how stable those two sets of clues are. Take a look at the top chart. That's the metabolites, the what's happening now data. See how a bunch of those blue bars are pushed all the way up to 100%? That means those signals are rock solid, super reliable. But now, look at the bottom chart. That's the gene data. It's a mess, right? The signals are all over the place, totally inconsistent. This was a massive hint for the researchers. The what's happening now clues were way more important than the what's the plan clues.
So after digging through all that complex, real-time chemical data, the AI's investigation didn't point to a whole bunch of different suspects. Nope, it all came down to one, just one single thing.
And that one thing, the single most important molecule, the smoking gun that could predict if the cooling therapy was going to work, was an amino acid called L-methionine. And this chart right here shows you just how unbelievably powerful that one clue was. Look over on the far left where the model is only using a single feature. Just L-methionine, all by itself, was enough for a perfect classification. I mean, 100% accuracy in predicting whether the brain was in a protective state or not. That is an incredibly clear signal to get from such a complicated biological system. Okay, so the metabolite clue? That was a total solo superstar. But what about the other side of the investigation? You know, the genetic plan? Well, Cosmos found that over there, it wasn't about one single superstar gene. No, it was about finding a small, elite team that all worked together. So how did it find this team? It used this really clever technique, basically removing potential gene clues one by one and seeing what happened. And as you can see right here, the model's performance stayed really high as long as it had 5 genes. But the second it dropped down to 4, bam, the accuracy just fell off a cliff. And that told them everything. A core team of exactly 5 genes was absolutely essential.
And this is what it found. The 5-gene conspiracy, if you will. TEX 19.2, MDM4, HTATIP2, Serbp1 and TMEM53. These 5 working together were the absolute bare minimum genetic signature you needed to track the brain's protective response.
But what's really cool is that this team doesn't just work all at once. It's actually more like a relay race. Look here. Right after the injury, at hour 0, Serbp1 is the key player. Then, it passes the baton. At 8 hours, HTAP2 takes the lead.
And by the 24-hour mark, Tex 19.2 is the one sprinting for the finish line. So each gene has its own specific time to shine, passing that baton of protection over a full 24 hours. Okay, but this led to one last big piece of the puzzle. The AI had found this super powerful metabolite clue, right? And it had found this 5-gene relay team. The problem was, they seemed to come from two completely different worlds. They looked totally unrelated. The what's happening now and the what's the plan just didn't seem to be talking to each other. So the big question was, how on earth are these two things connected?
And the problem was really clear when they looked at the data this way. On the left, you've got 122 biological pathways, all connected to those metabolites. On the right, 16 pathways connected to the gene signature. And in the middle, where they should overlap, nothing, zero. There was no direct connection at all. The two worlds were statistically separate, which just made the mystery even deeper.
The solution they came up with was just brilliant. Instead of trying to force a link that wasn't there, they built a kind of master detective. It's called a stacked classifier, and you can see how it works here. Basically, you have two specialists. One is the metabolomics expert. It only listens to our superstar L-methionine. The other is the transcriptomics expert, and it only listens to that 5-gene team. Then, a metaclassifier sits on top, takes the reports from both specialists, and makes one final, super accurate call.
And it was this master detective that finally revealed the hidden connection. If you look at this heat map, focus on that gene, H type 2. See that dark red box? That shows it's really strongly linked to something called glutathione metabolism, which is a key process for protecting cells from damage. And guess what? That process is directly tied to our star metabolite, L-methionine. And there it was. That was the bridge. The thing connecting the two worlds all along.
So, after all that incredible work, sifting through all the clues, finding the superstar metabolite, identifying the gene relay team, and building that master detective, what was the final conclusion? What did it all mean?
Well, the investigation could finally give a verdict on the original scientific hypotheses. The data absolutely supported the idea that these protective states involve changes in specific amino acids and that they avoid a harmful buildup of a substance called succinate. But, interestingly, the AI did not find evidence for other ideas, like broad changes to metabolic pathways or the brain using more fatty acids for energy. Basically, the AI helped focus the science on what was really going on.
And really, the final result just speaks for itself. The researchers put it perfectly. By combining that single metabolite, L-methionine, with the 5-gene panel, their final model could predict a neuroprotective state with over 90% accuracy. Think about that. They took two sets of clues that seemed to have nothing to do with each other and turned them into this incredibly powerful predictive tool.
So let's bring it all back to where we started. Remember that 50-50 coin flip? What if you could replace that with a prediction that's over 90% accurate? This discovery, it's not just about solving some cool biological puzzle. It's about opening the door to a future where doctors can know almost immediately which newborns aren't responding to that cooling therapy, and then they can give them the targeted, personalized help they actually need. It's about turning a coin flip into a life-saving, targeted intervention.